Hey Team Bio, welcome to your screencast about DNA, an introduction to DNA. So we're talking about deoxyribonucleic acid. This gives us our acronym DNA. We've been talking a lot in class about evolution and things being genetically encoded. And you might be wondering, well, what does that actually mean? This screencast is going to answer those burning questions for you. Okay, in biology, we have something that we call the central dogma of biology. And dogma is actually not a great word for it because it means it's like something that you do not question and you follow blindly. And in science, we should question everything, but whatever, that's what it's called, the central dogma of biology. It's basically talking or telling us the flow of information in all living systems. So living systems all store their information as DNA. This DNA is then transcribed into RNA, RNA, which is then translated into protein. And protein is what actually gives us our traits and characteristics. But it's the DNA that holds the information. So if I were to make an analogy, it would be like the DNA is the architect's blueprint, the master blueprint. And the RNA is like the copies that you actually take to the job site. And then the protein is like the house that you build out of the information in the DNA. Okay, so let's talk a little bit about the structure of DNA and how it encodes its genetic information. So as you all know, here is a nucleus. Because we have a nucleus in this image, we know that this cell must be a eukaryotic cell. Great. So the DNA in all eukaryotic cells is housed in the nucleus. If this were a prokaryotic cell, there would be no nucleus, but there would still be DNA. It would just be loose in the nucleoid region of the cell. Okay, DNA is stored in these structures that are called chromosomes. And chromosomes are actually composed of DNA, so the strand, and then also a bunch of proteins called histones. The histones are kind of like a spool, and the DNA is kind of like a thread. So the combination of histones and DNA is called a nucleosome. And this structure just keeps the DNA organized in the nucleus because you have a huge amount of DNA in every cell. So the nucleosome just helps organize the DNA in the chromosomes. Okay, if we unravel a chromosome, we would see that along specific areas of the chromosome, we're going to find a gene. A gene is, as you remember from your screencast last night, it's a location on a chromosome. A gene is the location on the chromosome. And if we look at my DNA, so we take a chromosome out of one of my cells, and then we take a chromosome out of one of your cells, because we're the same species, we would share all the same genes. What would make us different is the alleles. An allele is the sequence of nucleotides. At this gene. Well, what is a nucleotide? A nucleotide is one of the big four organic compounds. They all have a common structure And I'm going to draw it out for you now. So you can draw a 5-membered ring, and then a line with a P off of it, and then another line with a box off of it. So this is a simplified model of our DNA nucleotide. So all DNA nucleotides share a similar structure. They all have a sugar group. If it's DNA, it's deoxyribose. If it's RNA, it's just ribose. More on that later. They all have a phosphate group. And they all have an area called a nitrogen base. In DNA, nitrogen bases come in 4 different flavors. It can be A, T, C, or G. And I'm going to color it in yellow, purple, red, and blue. Okay, so this is a nucleotide. When we string a bunch of nucleotides together, we get a long strand of DNA. So if you look at a long strand of DNA, you can pull out a single nucleotide. So you can see here we have the ribose or deoxyribose sugar, because this is DNA, not RNA. We have the phosphate group, and we have the nitrogen base. Okay, let's label the base pairs over here. This is the red. I gotta look back at my key. Is cytosine. The blue is guanine. The yellow is adenine. And the purple is thymine. Okay, something that's important in understanding DNA is that these bases always come in pairs. Cytosine always binds from one strand to the other strand to guanine. So if you have cytosine on one side, there's going to be guanine on the other, and vice versa. So you see up here, we have cytosine on one side, guanine on the other. Cytosine on one side, guanine on the other. And then adenine and thymine always bond together. So if you find thymine on one side, you're going to see adenine on the other. This is called complementary base pairing. I guess I should write that down. The fact that A binds to T and C binds to G, it's okay, so that's one feature of DNA. A always binds to C, and C always binds to G. I guess another feature of DNA is that the two sides of the molecule are what's called antiparallel. So if we look over here, we have a nucleotide like this. If we look on the other side, this is our nucleotide like that. So you can see that the phosphate group is on the other side of the sugar relative to this one, whose phosphate group is on this side. So this strand of DNA is running in this direction, and this strand of DNA is running in that direction. We call this feature anti-parallel. So the DNA is running in opposite directions, but parallel to one another, anti-parallel. Okay, so the center is our nitrogen bases, and the outside is our sugar and phosphate backbone. I guess the other thing that I should tell you is that the order of cytosine, guanines, adenines, and thymines is a code. So just like letters are kind of a code, if I write down the word cat, it means something to you. But if I rearrange the letters to write act, it means something different. And if I rearrange them again to write tack, I don't know, I think you actually spell tack with a K. But you get the point. It means something different. So the order of letters in these words, it conveys different meaning. The same is true of the order of nucleotides in a strand of DNA. A gene is going to be a spot on a chromosome, but the alleles, the sequence of nucleotides, determines what types of proteins are made from this gene. So if we change the sequence, if we change the letters A, T, C, or G, swap them or move them around in any way, then we change the protein that is created here. So when we talk about changes to the genetic code, we're talking about changes in the order of C's, A's, T's, and G's of nucleotides. Okay, much later on in the year, we are going to break the code. We're going to learn how to decipher the order of A's, C's, T's, and G's and figure out how to hack into DNA and then create a novel, or I guess I should make it sound more exciting. We're going to hack into the DNA and we're going to genetically engineer a new protein, which will be very cool. Okay, that's all.